Welcome to Herb Harmony, your sanctuary for all things flora. In today's episode, we delve into the fascinating world of plant breeding techniques. As we journey into this realm, we'll discover how nature and science intertwine to create the vibrant and diverse plant life we see around us. We'll explore the art and science behind cross-pollination, a technique that brings together the best genes from different plants. We'll dive into the world of hybridization, where new plant varieties are born. We'll look at mutation breeding, a method that introduces new traits into plants. We'll delve into genetic engineering, a technique that allows us to change the very genes of plants. We'll also explore in vitro fertilization, tissue culture, and marker-assisted selection, techniques that have transformed how we propagate and select plants. Stay tuned as we unravel the mysteries of the plant kingdom, and remember, don't forget to like and subscribe to our channel. First up, we have cross-pollination, a natural marvel that allows plants to share their best genes. It's like a botanical version of a swap meet, where plants exchange genetic material to create offspring with a mix of their best traits. Now imagine you're a plant breeder. You're not just waiting for nature to take its course, you're actively facilitating this genetic exchange. You're looking for specific traits to enhance, like resistance to pests, drought tolerance, or perhaps a particularly vibrant bloom. So how does cross-pollination work? Well, it's all about the birds and the bees, and sometimes the wind, too. These agents of nature carry pollen from one flower to another, transferring genetic material in the process. But as a plant breeder, you can also manually cross-pollinate plants. You might use a small brush to transfer pollen, or you might even remove the male parts of a flower to prevent self-pollination, ensuring that the plant breeds with another. Through cross-pollination, you can create a diverse array of plants, each carrying the best genes of their parents. This not only leads to greater variation, but also to plants that are more resilient, able to withstand pests, diseases, and changing climate conditions. It's a testament to nature's ingenuity and our own as we work with the natural world to create something truly extraordinary. With cross-pollination, plants truly share the wealth, passing on their best traits to future generations. Next, we explore hybridization, a technique that has given us many of our favorite plant varieties. Now you might be wondering, what is hybridization? Simply put, it's the process of combining the genetic material of two different plants to create a new plant with the best traits of both parents. Imagine it like a master chef creating a new recipe. They take the best ingredients from different dishes and combine them to create something new, exciting, and most importantly, delicious. Similarly, plant breeders select two plants with desirable traits and through hybridization, create a new plant that has the best of both worlds. Now let's dive a little deeper into the process. It starts with the pollination of one plant with the pollen of another. This could be done manually by a breeder or naturally by insects or wind. The fertilized plant then produces seeds which carry the combined genes of the parent plants. These seeds, when grown, result in a new plant variety. The beauty of hybridization is in its simplicity and the limitless possibilities it offers. From roses with unique colors to tomatoes that resist disease, hybridization has been responsible for some of the most incredible innovations in the plant world. Through hybridization, breeders create a cornucopia of new, exciting plant varieties to enjoy. So the next time you marvel at a unique plant variety, remember the magic of hybridization that helped bring it to life. Now, let's delve into the realm of mutation breeding, where new traits are born. Mutation breeding is an exciting technology that harnesses the power of nature to create plants with exceptional traits. Think of it as a shortcut to evolution, where we speed up the process of natural variation to bring about desirable changes. At its core, mutation breeding involves inducing changes in the plant's DNA. This is done using physical agents like radiation or chemical mutagens. These mutations or changes can lead to a variety of outcomes, from enhanced disease resistance to improved yield, the creation of novel colors, shapes, or even new flavors. But how does this process work? Well, once the mutations are induced, the next step is selection. This means growing the mutated plants and choosing those with beneficial traits. It's a bit like a botanical talent show where we pick the star performers and propagate them. While it might sound a bit like science fiction, mutation breeding has been used for over 70 years and has resulted in thousands of improved plant varieties. It's a powerful tool that helps us adapt to changing environments and meet the growing demands of our world. So, next time you bite into a ruby red grapefruit or admire the vibrant hues of a chrysanthemum, remember that these desirable traits might just be the result of mutation breeding. Mutation breeding, while sounding a bit sci-fi, is a real and impactful way to introduce new traits into our beloved plants. In the realm of genetic engineering, we wield the power to change the very genes of plants. This isn't science fiction, it's a reality that's revolutionizing the way we approach plant breeding and cultivation. Genetic engineering, in the simplest of terms, is the process of modifying an organism's genetic structure. In the world of plants, this means we can alter the genes in a way that brings about desired traits. This is like being an artist. With the plant genome as our canvas, we can introduce, enhance, or even eliminate certain traits, creating custom-made plants that are better suited to our needs. Let's take, for instance, a tomato plant. With genetic engineering, we could potentially create a variety that's resistant to certain pests or diseases, saving us the trouble of constant upkeep and pesticide use. Or perhaps we could engineer a strain that bears fruit all year round, regardless of the season. The process begins with identifying the genes responsible for the traits we wish to modify. Next, these genes are isolated and introduced into the plant genome using various techniques. It's a meticulous task, requiring precision and patience, but the results? Well, they're nothing short of extraordinary. Genetic engineering isn't just about creating super plants. It's a tool for sustainability, for resilience, for a future where our food is secure and our environment preserved. With the power of genetic engineering, the possibilities for plant improvement are virtually limitless. So the next time you admire a plant, remember, it could be a masterpiece of genetic artistry. In the world of in vitro fertilization, we can grow plants right in the lab. Imagine a future where we don't have to rely on the whims of nature to cultivate plants. A future where we can engineer the growth of plants right down to the cellular level. This is the fascinating realm of in vitro fertilization in plant breeding. In vitro fertilization, or IVF, is a process where plant embryos are formed outside the parent plant's body. It starts with the extraction of pollen and ovules from a parent plant. These are then brought together under controlled conditions where fertilization takes place. The resulting embryos are then transferred into a nutrient-rich culture medium that fosters their growth. But why do we need IVF in plant breeding? Well, it allows us to overcome barriers that exist in nature. It enables the fertilization of plants that might not naturally pollinate. It also allows us to control the genetic makeup of plants. In essence, it gives us the power to create plants that are more resistant to diseases, more adaptable to different climates, and even more nutritious. This is not science fiction, but a reality that's happening in labs around the world. Plant scientists are using IVF to create new and improved plant varieties that could potentially revolutionize agriculture. In vitro fertilization brings the miracle of plant growth right into the lab, opening up a world of possibilities. With tissue culture, even a tiny piece of plant tissue can give rise to a whole new plant. It's a fascinating process that demonstrates the remarkable potential of plants. At its core, tissue culture is a technique where a small piece of plant tissue is taken and placed in a sterile environment. This environment is often a nutrient-rich gel, providing all the necessary components for the plant tissue to grow and develop. This technique is akin to the concept of cloning, where the new plant is genetically identical to the parent plant. It's an incredibly effective way to propagate plants, especially those that are rare or difficult to grow from seed. What's more, tissue culture can help protect species from extinction by allowing for the mass production of plants in a controlled environment, free from pests and disease. Tissue culture truly showcases the resilience and adaptability of plants, capable of regrowth from just a tiny piece of tissue. Finally, we have marker-assisted selection, a technique that uses DNA markers to select the best plants. This method is like a high-tech treasure hunt, where the treasures are the desirable traits in plants, and the map is the plant's own DNA. Marker-assisted selection starts by identifying specific DNA sequences or markers linked to the traits we're after. These markers act like signposts, pointing us towards the genes responsible for traits like disease resistance, high yield, or superior flavor. The real beauty of this technique is that it allows us to screen a large number of plants quickly and accurately. We don't need to wait for the plant to grow or express the trait. The DNA markers give us the information we need right from the get-go. Marker-assisted selection takes the guesswork out of breeding, allowing us to pick the best of the bunch down to their DNA. As we conclude our journey through the world of plant breeding techniques, let's reflect on what we've discovered. From cross-pollinating to create plants with the best genes, hybridizing for new varieties, mutating for unique traits, and genetic engineering for targeted changes. We've seen the possibilities of lab-grown plants through in vitro fertilization, tissue culture propagation, and the precision of marker-assisted selection. Thanks for joining us today on Herb Harmony. Remember, the world of plants is full of wonders waiting to be discovered. Stay curious, keep exploring, and we'll see you in the